Hi, so welcome back. So in this lecture we're going to talk about how electrical signals actually are created and transmitted in neurons. So with that, hopefully I can get my slide to advance here. Alright, so let's just do a quick little recap of stuff we've already talked about. So basically the way things are going to work, again, so we have this sensory neuron. I think I mentioned this example to you before, where you get the sensation of actually holding your pen. And that's going to send the signal up to the brain, and you can see here's the cell body of that sensory neuron way in the spinal cord. That's really impressive that basically the receptor part is here. It'll travel all the way up to the spinal cord, and then from there it's going to go up to the brain, to a part of your cerebral cortex. We'll talk about that more when we get to the brain. It's going to take that information, integrate it, send the correct information back down through your spinal cord to the muscles, and in this case, telling your hand, okay, write whatever letters it is you are writing when you're taking notes. Okay? So what we're going to talk about now is how do we get this signal? How does it go all the way from here, go all the way up to the brain? Because that's kind of interesting, right? Like, well, how does that work? All right. So again, let's just kind of take quick reminder of our neuron. So here we have our dendrites. So they're going to bring in the incoming information. Here's the cell body, the soma or the parakaryon. It's going to integrate some of that information, and then it's going to get transmitted. You can see these arrows down the length of this axon to these axon terminals, to these what are known as synaptic end bulbs at the terminals. So they're going to release neurotransmitters. Okay? So this is what we're talking about. Like, how does this actually happen? Okay. So when we're going to talk about this, what we're going to need to talk about is really going to be two key concepts. I can hide me here for you. And try to get my slides advanced. There we go. So some definitions. We have to understand these before we can go any further. So the measure of potential energy, so remember potential energy, that's energy that sort of has the potential of doing work. So that potential energy is going to be generated, so created, by a separated charge, and it's called voltage. In our case, we measure it in millivolts. When we talk about our batteries, you know this as well. I mean, you have a 9-volt battery, you have a blah, blah, volt battery, right? And just like batteries, we have a separation in charge. Your battery has a positive end and a negative end, or a positive terminal and a negative terminal, right? So we've separated charge in batteries, and when we hook them up, we have voltage. Well, basically we have voltage by that separated charge. When we hook them up, what's going to happen is we're going to have a flow of electrical charge, and that's how we get a current. And really, this is what we're going to care about, is how do we actually get these electrical charges to flow from one point to another, so that we actually can send a signal. All right. So what I'm going to talk to you guys about now, I have a blank slide coming up so I can draw this for you, is basically I'm going to talk to you about how we actually separate the charge, because that's the number one thing, and then how do we actually, once that charge is separated, how we're actually going to get it to move from one place to another. Okay. So with that, try to advance my slide here. Bear with me, because I'm going to be doing quite a bit of drawing. So when we're talking about voltage, what's going to be separating our voltage is going to be the cell membrane. So here's the cell membrane. So here's one part of the so this is the phospholipid bilayer. So this is outside the cell. Okay, and this is inside the cell. And by inside the cell and outside the cell, we're really talking about the axon. So this, so here's the other side. This whole thing would be the axon. It's really long. This is just a super zoomed in image. Okay. So I'm just going to concentrate. Let's see, pen color. So what we're going to do is we're going to have different channels. Remember, like way back at the beginning of the semester, we had talked a lot about the sodium-potassium pump. And we talked about how it pumped a lot of sodium ions outside the cell, right? And a lot of potassium ions inside the cell. That's what sodium-potassium pump did. And if we gave a channel in that cell membrane, potassium would want to leave, go from where it's highly concentrated to where it's less concentrated. You guys vaguely remember that, hopefully. So what you're going to find is that your cell membrane actually has what is known as a potassium leakage channel. So this right here is a potassium leakage channel. And basically what it's doing is it's allowing potassium to leak out. Okay? So this allows potassium to leak out. That's what a leakage channel does. But let me be very clear. Just like when we say something is leaking, like a faucet is leaking, it's not a giant flow of water, right, in the faucet. It's sort of a drip. Similar here. So it's essentially like a little bit of potassium is leaving the cell. However, you still have a much higher concentration inside the cell than outside. Okay, hopefully that makes sense. Now, as this potassium is leaving, right, basically what we have is a positive charge is leaving. It's going this way. Right, you following me so far? Okay, now you have lots of other charged ions inside the cell. So as this positive charge is leaving the cell, basically following right behind it like a little puppy dog are going to be these negatively charged ions. Because again, positive and negatives attract. But there is no ion channel for the negative charges to get out. So the negative charges end up lining up along the inside of the cell membrane because all these positive charges, they're attracted to these positive charges out here that have leaked out. Okay. Now, if you get enough of these negative charges lining up on the inside of the cell membrane, these positive charges will be attracted back to them. Okay, but they can't get back in. These leakage channels, just like your faucet, the water drips out of it, doesn't go back up it, they can't get back inside the cell. Okay, because again, they have to have a special channel that would allow them back in. So again, potassium ions, again, more highly concentrated inside the cell than outside, thanks to our sodium-potassium pump. We put in leakage channels into the cell membrane, which allow them to leak, again, drip, 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 kind of out. That causes negative ions to want to follow them. However, the negative ions cannot get out, so they line up along the inside part of the cell membrane. That then causes the positive charges outside the cell membrane to also be attracted back across, but they can't get back in, so they line up along the outside of the cell membrane. So what we have done here with this leakage channel is we've created a separation in charge, right? Positives are outside, negatives are inside. Since we've separated these charges, we now have voltage. All right. And in fact, if I were to take, and they do make these, it's incredible how they make them. They take like little electrodes. You can actually, it's tiny. They're tiny, tiny little electrode. So you stick an electrode right like inside the cell membrane of this axon to measure the charge difference. So you've got like a little machine that measures this, right? There's a machine that's measuring things. What you'll find is that the inside of the cell membrane is at negative 70 millivolts. So it has negative 70, like it has a negative charge of all these. It's negative 70 millivolts compared to the outside of the cell membrane. Okay. So hopefully that makes sense. So we have these and again, batteries, we're talking volts, 9-volt battery, whatever voltage battery you have. I mean, these are millivolts, so 1,000th of that. Okay, so we don't have that same kind of voltage that a battery will. However, it does exist, and we can actually measure it. So when a cell is at rest, so let me just kind of graph this for you. Here's our graph. So let's say here's voltage on the y-axis. So at rest, So let's say here, let's say this is 0. So down here is negative 70 millivolts. You put an electrode in, you'd see that that cell, when it's at rest, at that axon, it's just kind of right there at negative 70 millivolts. That's the separation in charge. All right? Now that's still not the electrical signal. All we've done is separate the charges. This will now lead to the electrical signal. So let's see that. Okay, let's see. Try to clear this. Okay, actually, you know what I'm going to do? So I'm going to stop the recording right here. And I'll record how we get our current in the next one. So it's a nice smaller snippet. So if you need to like just refer back to this, you can. Okay. So again, what we talked about now is how we separated the charges and created voltage. The next lecture is going to talk about how we then get these ions to move, and therefore we create a current. And by creating a current, we're actually sending that electrical signal down the length of the cell membrane to another cell or another tissue. Okay. So let me know if you have any questions, and go on to that next recording.